A very good evening, students. So, as you know that we have started with our revision work from our topic on the enzymes from the chapter biomolecules, and today we will talk about the last portion of enzymes, which is on our page number 158 and 159 of NCRT. So I will request all of you to go through this topic from NCRT also, and I will tell you some tricks to learn this, to write this with the efficiency, and to attempt the questions in a very short time with these tricks. So, when we talk about the nomenclature, the scientist name which came in the picture, that is Doclex. Doclex in 1883 he talked about the nomenclature of enzymes, and if we talk about the very basic naming, that how do you give names, how do you recognize that this is an enzyme, if you recall, we usually use the word ase at the last in any enzyme. Suppose I have to digest carbohydrates, so the enzyme which is used there is carbohydrases. You are going to digest proteins, the enzymes we use is proteases. You are going to break down ester bond, then there will be esterases. So as you can say, this ase word is very common, basic, simple nomenclature with the enzymes. Now, if we talk about the classification, that how can you classify enzymes, there are very six very famous, very known categories of different enzymes which are included in our syllabus. That is six categories, and but how to remember them, that is a major concern. How to remember all these in the during the exam, and plus this question comes in 5 marks during exam, and we have to write down the examples also. So there is one trick, always remember a one line, that is over the hill, right? And what are you going to take from this word now? Over the hill. We are going to take these six letters, 1, 2, 3, 4, 5, 6 letters. They are going to be in use for us, right? So first category is oxidoreductases, second is transferases, right? So in this way you can write down this efficiently, as I have written here, over the hill, over the hill. So this is the mnemonics to remember this six categories of enzyme during your exam time: oxidoreductases, transferases, hydrolases, isomerases, lysases, and ligases. Right? Now the comes here is first category of enzymes, that is oxidoreductases. बहुत बच्चे गलती करते हैं यहाँ पे oxidoreductases, and this also include the example of dehydrogenases. See, oxidation and reduction, three categories are there, in subcategories are there in oxidoreductases: oxidases, dehydrogenases, and reductases. But before you can classify, you can define this category, you need to understand what is oxidation. लूज करना, राइट? मतलब मेरे पास ऑलरेडी बहुत कुछ है, बहुत ज़्यादा है, लॉस देना, लॉस ऑफ इलेक्ट्रॉन्स, एंड समटाइम्स वी से गेन ऑफ ऑक्सीजन, क्योंकि ऑक्सीडेशन है, सो गेन ऑफ ऑक्सीजन, और वी कैन से लॉस ऑफ hydrogen when they transfer, when they give hydrogen, actually not, I will not use the word transfer, they give hydrogen. So loss of electrons and hydrogen, or you can say gain of oxygen. This is the definition of your oxidation. And those enzymes which catalyze these reaction in which loss of electrons is there, that are called oxidases, right? रिडक्टेशन इज टोटली ऑपोजिट ऑफ ऑक्सीडेशन. अब आपने लॉस किया है, लूज किया है ऑक्सीडेशन में. सो रिडक्शन मीन्स गेन ऑफ इलेक्ट्रॉन्स, गेन ऑफ हाइड्रोजन. बट यहाँ पे यू आर गेनिंग ऑक्सीजन, हेयर यू आर लूजिंग ऑक्सीजन. सो एंजाइम्स विच कैटालाइज दीज रिएक्शन गेन ऑफ इलेक्ट्रॉन, दे आर reductases. Addition of hydrogen. Oxidases, what are you losing here? Loss of hydrogen. When you are losing your hydrogen, it means it is transferred to something else. So hydrogen is shifted to form a molecule, that is to form a molecule to oxygen, basically from a molecule to oxygen. So hydrogen is shifted here. The enzymes which shift, which lose this hydrogen, they will be oxidases. Very, very, very important part. These oxidases, they play a role in photosynthesis, because we know that oxygen is produced there in respiration, then water is released there. So hydrogen shifting is very, very important, which is catalyzed by oxidases. So examples, which examples? ऑक्सीजन का नेम आते ही फोटोसिंथेसिस एंड रेस्पिरेशन. इट इज़ अ वेरी इंपॉर्टेंट एंजाइम्स विच प्ले रोल इन ईटीएस, इन फोटोसिंथेसिस and respiration. Now comes to, if one is giving hydrogen, another is adding hydrogen. So addition of hydrogen or addition of electrons, or you can say removal of oxygen, that is your enzyme nitrate reductase. It works only in the presence of coenzyme helper, that is called NAD, nitrate reductase, which play a role in addition of hydrogen. Basically during this, hydrogen is added, electrons are added. So in these kind of enzymes in which hydrogen is added, electrons are moving, adding, these are reductases. Next is dehydrogenases. Dehydrogenases means you are transferring hydrogen, but transfer किसको कर रहे हो? Not to the any molecule. You are adding hydrogen is transferred usually to the coenzymes. Coenzymes are NAD positive, NADP. These are coenzymes. So succinate dehydrogenase, this is the very common example, as well as oxidation of molecules. Oxidation of molecules. Oxidation of molecules means now losing hydrogen during aerobic respiration. So dehydrogenases and oxidases, they usually work together in the respiration and photosynthesis. Dehydrogenase specifically works for the coenzymes. So three categories in oxidoreductases: oxidases, reductases and dehydrogenases. Or simply you can write down in your exam that these are the enzymes which play role in oxidation and reduction reaction. Fine, these are those enzymes. Fine. Next is the transferases, over the hill. So transferases, transfer karna, they help in transfer of a specific group. It can be methyl, it can be amino group, it can be acyl. So you are transferred to a king's place. Mostly this category include the example of groups, like you are transferring amino group, transaminases, you are transferring methyl group, methylases, you are transferring phosphate group, phosphorylases. Then king's place, kinases. Kinases are very important enzymes which release phosphate on breaking down of ATP. When ATP is breaking down, then one phosphate is released. So kinases and transphosphorylases are very similar. Only difference is it does not include the ATP breakdown, but kinases mostly include the breakdown of ATP during the catalyzation of reactions or during the phosphate transfer. So transfer जब कोई group transfer हो रहा है बच्चा, that those are the category of transferases. You are adding methyl group to a substrate, you are adding amino group to a substrate, you are adding acyl group to a substrate, so you are transferred to King's place. Always you will be able to remember kinases and transferases, transaminases, methylases or phosphorylases. Hill, Hill. So Hill, first H, hydrolases, hydro, water. These help to catalyze your food. Breakdown of food. Breakdown of food. What is food? It usually include large molecules. Either you talk about carbohydrate, it is a big molecule. Then you talk about protein, it is a larger molecule, macromolecule. Then you talk about your fats, that is a large molecule. So breakdown of large molecule, but in the presence of water. By addition of water, only you can break down your organic food. And mostly very strong digestive enzymes are present in our one of the organelle, that is lysosomes. Lysosomes have very strong digestive enzymes. So all the digestive enzymes, like either you talk about lipases, amylases, carbohydrases, proteases, esterases, they help in breakdown of your glucose, they help in breakdown of proteins, they help in breakdown of starch, or you can say ester, or you can say fats. So basically, but in the addition of water, or you can say in the presence of water. So hydro word aate hi, they categorize, basically they catalyze large molecules. I will use large molecules there. So you can mention any category. Food कोई भी दिमाग आपके brain में आता है तो you can mention these categories, or you can learn this trick. There is a one festival in our foreign country, that is Easter day. They celebrate Easter day. On Easter you ate a lot of food. So food, say you can remember carbohydrates, proteins, fats and water. Water is very important in hydrolases. Now next come is liases. Liases break down specific bonds, as well as it removes group. So but it does not use water. It is without addition of water. And very good example of this is aldolases. Aldo is a shoe brand, so you can remember from that aldolases and boxing glove, decarboxylases, boxing, decarboxylases. These are the example of liases. So liases are enzymes which break covalent bonds, which remove groups. Very important example here is histidine. Histidine is an amino acid. Histidine, when on histidine there is an enzyme which acts, that is decarboxylase, histidine decarboxylase. These, it removes this carbon-carbon bond. It break this carbon-carbon bond and remove this group and change into histamine. Histamine is released during the allergic reaction by the mast cells and basophils. So this is the example of your liases. Decarboxylases, aldolases, these are our liases, in which breakdown happens, definitely breakdown happens, but in the absence of water. So here one major thing which is formed after removal of this group, that is the CO2. Now another form is Isomerases. When we say isomer, like glucose isomer is isomeric form is fructose. It means they are similar. They both are 6 carbon. They both are 6 carbon compounds. The only difference is in their structure. The only difference is arrangement of their structure, right? So when there is a rearrangement of the molecular structure to form the isomers, that is done by the enzyme isomerases, like your phosphoglucomutase, phosphohexose isomerase. Suppose now I'm making glucose 6 phosphate. I already have in my hand glucose 6 phosphate. I want its isomeric form, fructose 6 phosphate. What will I add? I will add isomerase. I will add isomerase, right? So basically they act on glucose 6 phosphate and it will form fructose 6 phosphate. You are not adding any phosphate group here. You are only arranging the structure. As you can see, that is a deoxy sugar and this is a ribose sugar. So only difference is in their structural formula. Otherwise their chemical formula is same. Both are 6 carbon sugars. So when we are only rearranging the ऑप्टिकल फॉर्म्स और आई विल से स्ट्रक्चरल फॉर्म, जब भी कभी आप स्ट्रक्चरल का रीअरेंजमेंट कर रहे हो देन यू विल यूज आइसोमेरेजेस. एग्जाम्पल विच यू कैन राइट डाउन, ग्लूकोज 6 phosphate to fructose 6 phosphate, phosphohexose isomerase, राइट. 6 कार्बन है और हम सिर्फ आइसोमेरिक फॉर्म में चेंज कर रहे हैं एंड द एंजाइम वी आर यूजिंग आइसोमेरेज. Last one is ligases or synthetases. Ligase, they work like your Fevicol. They work as a glue. They help in joining two molecules together. So basically, how do you join two molecules? I have to join this A plus B. I have to join them. I have to form this बॉन्ड. सो बेसिकली दे फॉर्म बॉन्ड्स. अब तक हम तोड़ने का काम कर रहे थे, वी वर ब्रेकिंग डाउन द बॉन्ड्स. दीज आर द एंजाइम्स विच फॉर्म बॉन्ड. हाउ विल यू फॉर्म बॉन्ड? You need something to form a bond. So they use energy from the ATP. DNA ligase is very best example of these category is DNA ligases. They break, basically they use to repair, they join DNA molecule. Citric acid synthetase, because these ligases are also known as your synthetases. In this category there is a synthetases also come. So citric acid synthetase, usually what happen is citric acid, जब आप बहुत when you consume lot of citric acid you may sometime feel gastric problems, right? So from gas you can remember that this citric acid synthetase is example of ligases or synthetases, right? So citric acid synthetase is example of ligases or synthetases, as well as you can mention in your exam DNA ligases. Join karna, just remember, like a Fevicol, they form bonds. So this is all about your classification of enzymes. The last thing which I have to discuss with you, that I will discuss about the cofactors in the another video.